Switching over. Good. Looks like we're right. Looks like we're live. Yeah. I have a live feed so we can see and make sure everything looks good. And yeah, we're live. Look at that. All right. Well, welcome everyone in. We're at the Frozen Zoo today, and we have two very special guests from the zoo. We're celebrating 50th anniversary. Would you like to introduce yourselves?
Yeah. Thank you very much. Happy to be here. And by the way, a little slight correction too, because we're San Diego Zoo Wildlife Alliance as a whole is the name of our organization, but we have two conservation parks here in sunny San Diego. So the San Diego Zoo is actually in the heart of San Diego, downtown. We're talking polar bear, we're talking giant panda, but here is where our favorite place, I would say, right? The San Diego Zoo Safari Park. So we're in the town of Escondido, the North County of San Diego. And this park is 1,800 acres, and I get easily distracted because of the hummingbird that just perches right above you, which is the perfect example of all the wildlife that we have here. It's over 680 animals. We're representing roughly around 350 species and about 1.3 million plants that live here at the safari park. And my name's Marco, by the way. I'm the spokesperson for San Diego Zoo Wildlife Alliance, but we have someone even more special than I, the curator of the coldest zoo in the entire world. So, amiga, you want to introduce yourself, Laura?
Sure. I'm Marlys Houck. I'm the curator of the Frozen Zoo, as Marco said, and I'm not the one who usually is our public speaker, so Marco is going to be a great help today.
I'll do my best. I'll do my best to represent. But it's the diversity of people that highlight all the conservation work that we do here, right? That's what it's about.
Thank you all so much for joining us today, for letting us come in the behind the scenes tour. It's a dream come true for many of us to be able to see this place. Give us an idea of, you hear Frozen Zoo, and like you said, it's the world's coldest zoo. So what makes it for the Frozen Zoo? What is here about this center that's so special?
Ooh.
So the Frozen Zoo is the largest collection of its kind. What's really unique about it is its living cell lines, not collection of DNA. There is DNA inside of each of the cells, but the living cells they can be pulled out and grown again, expanded. But we have the largest collection. It's birds, mammals, reptiles, amphibians, and a couple fish. So it's all vertebrates. And they represent right now we have over 11,600 individuals, and it's always growing.
Wow.
We have over 1,300 species and subspecies in the Frozen Zoo over this 50 years. So it's a remarkable collection, and there's none like it in the world.
There was we actually interviewed Marlys here for our podcast called Amazing Wildlife, by the way. And you said a really cool fact during the interview that you had said that the amount of wildlife represented in your Frozen Zoo,
right,
it's nearly double the amount of animals at the park and the zoo combined, right?
Yeah, right.
That's incredible,
right.
The diversity
and
of it.
The reason is because if you think about the animals at the zoo or the safari park, they live a lifespan and then they die. So over 50 years, there aren't that many species that live 50 years. But in the Frozen Zoo, we bank the cells and retain them in there. And they never go away because we don't want anything to thaw. So the cells are always in there, and it's just continually growing.
So what can one do with a cell? If you take a skin cell from an animal, how is that going to help us save this animal and this species in the long term?
Right. So we have a lot of methods. There's a lot of ways for genetic rescue. I mean, one of the biggest things is it's a bank of genetic diversity. So every individual's genetic diversity is banked in there. And so when populations get to a really small number and they're inbred, there are ways to use that genetic diversity. One is through cloning. You can pull out a cell and clone an individual. And if that cloned individual reproduces, then that genetic diversity is put back into that otherwise tiny population. Because they're living cells, they can also be reprogrammed into stem cells. And then stem cells are that early cell form that can become any cell in the body. We've had some of our rhino cells, our stem cell team has reprogrammed them into cardiac cells that actually you can see them beating.
Wow.
But one way you could do genetic rescue with that is the cells could technically be turned into sperm and eggs. And so then you're back to everybody kind of understands how sperm and eggs can save a species. And the part that I'm over are the living skin cells, but there's also another half of the Frozen Zoo collection that is sperm and eggs and gametes. But I don't talk about that as much. It's not my specialty. It's all under Dr. Barbara Durant. But I think people understand intuitively why you would collect sperm and eggs, but not so much the skin cells.
Yeah, I mean, it was fascinating to me, I have to say. I'm definitely not from a science background, more animal behavior, but talking to Marlys here, I'm learning that from a wildlife care specialist aspect, you come to a zoo, we have our bird department, reptile department, mammal department, but it's almost a similar parallel. They have different specialties and skill sets depending on the species or wildlife that you're representing. Wouldn't you say?
Exactly. They're called the gamete team or the frozen zoo team. We don't know how to freeze gametes, and they don't know how to grow and freeze cells. The gametes, you get them, say sperm, and you can freeze it right away. The skin cells have to grow in an incubator and divide until there's enough to put in the frozen zoo. And that can take a month. If it's an amphibian, it can take 2 months to 6 months. Some are more difficult than others.
And there have been instances that we've seen on YouTube where it's like the last animal that y'all have gotten a sample of, and there's like the one left, and one of your team members are put in charge of growing up those cell lines. Can you talk about like what is the stress of that, right? Like you have the last animal cells there.
Well, I can tell you about there was a bird, it went extinct, called the pauli. It's a Hawaiian songbird, and the last one left they had only been rediscovered, I think, in the 90s, and they were trying to bring them into managed care together. They thought there might have been 3, kind of like the success we had with the California condors when it was down to a really small population. They brought them into managed care, and they were able to grow the population to what it is today and re-release them back into the wild. The pauli, there weren't enough left, and the last individual died, and it was Thanksgiving weekend, of course.
Yeah.
And the body was shipped here because we have a collaboration with the Hawaiian team. And they said, you got to try to grow cells from this. And I had said, you need to send it to 2 places so we can all try. But there wasn't anywhere else that was doing it at the time, which is one of the reasons we want to teach our methods to other people and get this set up around the globe. We need people with these skills all over the world so we don't have to ship samples. We have to get the sample within 2 or 3 days after death to get the cells to grow. And at the time, we were only doing mammals. The collection, it was started, as you said, 50 years ago by Dr. Kurt Benershka in 1975, and he was only doing mammals at the time. And one of the other reasons, you asked what cells can be used for, the live cells. When a cell is growing and dividing, you can look at the chromosomes. And he was really interested in the different chromosome numbers of animals. They range from, in the mammals, from 6 to, now we just recently learned, 114 in the white-bellied pangolin, an African species of pangolin. And so anyway, he was studying the chromosomes, and mammals was enough to keep everybody busy. But we had been asked to figure out the sex of the California condors so that when they paired them up, because isn't it true they kind of pair for life?
Yeah. So it speaks to the nuances of all the species that we have, whether it be a Przewalski's horse, a taki, they're known regionally, or a California condor. So it's not only those things. You have to think about these species-specific behaviors. And to your point, yes, they are bonded for a very, very long period of their entire lifespan. So that's something you have to account for, right? In your field.
Right. And so one way to look at the chromosomes from birds was to get growing feathers. When the feather is growing, there's pulp inside, and we can extract the pulp and grow cells. So we had some experience with growing feathers of birds, but what are the odds that this little pauli, they're the size of a sparrow, I think.
Yeah.
And he's our bird guy. So when I had done some sexing from feathers, but never had I grown cells from the bird. We knew the skin didn't work very well.
Yeah.
So here comes this bird, and they're like, if you don't freeze it, if you can't grow cells and freeze it, we lose that opportunity. I mean,
how does that feel when you're presented with this? You are the last person who can save this species' genetic information.
It was really stressful. It was kind of surreal. There were only 3 of us there that day. The head pathologist and our I think he was a curator of birds who was working in Hawaii, Alan Lieberman.
Yeah.
And it was just the 3 of us on this weird day after Thanksgiving, quietness. And
Everyone else is digesting their food, but not you guys.
yeah.
They got to be sharp on the ball. We're talking saving species here.
Right. And there was a delay getting the body in, so we had a lot of time to sit and contemplate. And it was surreal. And then, you know, we hadn't done it before. If it didn't have feathers growing, and it didn't have feathers growing. So what happened is I built on the knowledge that came before me. Dr. Benershka and Arlene Kumamoto, who had grown eye cells of she had used the cornea to grow whales and to grow one whale. I saw in the books that there was whale cornea. So I thought, well, maybe I can get the cornea and try that. But the eye was the size of a blueberry, so I couldn't really extract it out. So I just did, and I told you I have very scientific words: front of eye and back of eye.
Good to know.
And front of eye is where the cornea would be. But it turned out all the other things I set up as well: liver, kidneys, spleen, skin, gonads, nothing else grew, but the back of the eye grew.
Wow.
So today we know, we do a lot of birds today. Now we've expanded, and eye is one of our favorite tissues, and trachea. So these are tissues that repair quickly if they're damaged. Say if you injure your eye and you're in a wild species, your eye has to repair quickly so you can see, or your trachea has to repair quickly so you can eat. And had I known that the trachea would work back then, I wouldn't have been as terrified. But this was new territory, you know?
Yeah.
So it was terrifying. And then
presumably we didn't know why the eye would work, right? We just had a guess that it worked in this other instance.
Right.
Yeah.
Right. And pretty much you should be able to grow cells from anywhere in the body, but it doesn't always work. So
and even like the cell culture media. So the juices that you're growing the cells in can vary between species and what requirements they need.
For sure.
Right. So you're having to account for all these variables and all these tissues with this one small animal that's on its way, and it's delayed.
Right.
So you're also losing time on that front to be able to get your hands on work in there.
Right.
Yeah. I mean, you think about like when she was telling me this story, I thought, what a Wild West scenario, right? That, you know, especially in the world that we live in today with media and TikToks and all the things that's accessible to us at our fingertips. But there's so much about Mother Nature that we don't know about. And these guys are just doing cutting edge, like completely brand new stuff. And talking about this one species of bird that we're talking, native islands, they're losing so much biodiversity and wildlife. So we're not only doing work here, say with a polar bear, with an African elephant, but even a very important species of bird that can be found in one specific island or just several. So it just speaks to the importance of the work that you're doing. And you were telling me before it was eye and trachea for bird, but then for reptiles or amphibians, it was different.
Right. And so these we only do animals after they die. Mammals, we can do like a little skin biopsy, just like my dermatologist takes off of me. But birds and reptiles, amphibians, we wait until they die. And so what was your question?
It was tongue. Right?
Oh yeah,
amphibian.
We like tongue and foot.
Right? Yeah, I love that.
Yeah,
the nuances of it.
And that's presumably also because of cell turnover. So there's like the cells are predisposed to be able to want to grow in a dish, or like the conditions.
Right. It's not natural for them to grow in a dish. So we're forcing them to do this. And we have to have, as you said, the right media, the right reagents, the right temperature. Amphibians, for example, sometimes they grow best at room temperature. We have, our incubators are set in Celsius, and the incubator is 27 for amphibians. Room temperature is about 22, 23. And it depends, you know, if they're a tropical amphibian at the warmer temperature, if they're living in cool creeks like the mountain yellow-legged frog we have here, that would go at the room temperature, which is cooler.
It's really interesting how you're wearing so many hats when setting up this experiment, right? It's not just a molecular biology background, but you're taking into the ecology background, right? And presumably evolutionary background too, to be able to make the best guesswork of what's going to work, especially in the instance of the bird. And that did have a happy ending, right?
It did have a happy ending, And it takes a while. So what we do is we take that tissue, we dice it really small, we digest it in an enzyme, and the cells will, in theory, come out and attach to the bottom of the flask, so they're adherent. But that takes some time. And I think in this case, it might have been almost a week before I began to see cells. It was an older individual, just like if you think about humans, if a baby has a cut, it heals quicker, whereas an older person, it heals more slowly. The cells don't divide as quickly, and that happened with this bird. It was slower, and it was pretty agonizing waiting.
Like, when do you know that it's like oh no, we haven't worked. There's still hope. There's still time, and right, each animal's different.
Right.
So how do you know when the cutoff is? But how was that day when you saw them dividing? You're like, it worked.
Well, it was great, but I wouldn't say it worked. In fact, we don't even mention it. We're like, things are seeming good. We don't want to booger it. It's like, you don't want to, things can still go wrong. They could get contaminated. The cells could stop growing and dividing if they don't like the conditions. So you're never really sure until, for me, it's when it goes into the frozen zoo, the day that we get it in the frozen zoo. And then we freeze the files that we can use some for different things, and we pull one out to check viability. We don't just say, oh, I froze it, therefore it exists. I froze it, will it recover from the freeze? So we'll always pull one out. And we got to look at the chromosomes. No one had ever seen the chromosomes of a pauli before, the cells of a pauli. I mean, freezing that one individual will not bring back the species, not now anyway, I don't know, about 50 years from now. But learn its genome. The cells can be used for genome sequencing. It's one of the preferred materials because it's so fresh and not degraded DNA. We look at the chromosomes. We couldn't do that with the Tasmanian tiger or the passenger pigeon or the dodo bird.
And those specimens were from like museum samples, right, that had broken down over the course of time. So like you said, not as fresh, not as reliable, but you have it at least parts of the genome.
You could do the genome sequencing of those, but you couldn't look at the chromosomes.
Exactly.
But Marlys, you said something I think it's key. The idea of we don't know what's going to happen now, but maybe 50 years from now. And that was a core of Dr. Bernushka as well. Like, we don't know why we're collecting. He wasn't quite sure why we're collecting as much information as we're doing at the time, just because he didn't have the tech during that period in history, but now it's just further along and along. So I love the concept of for what we don't know yet, but we're going to preserve it.
Right. Yeah.
Well, I feel like no one would have thought that you could have taken skin cells 50 years ago and made an embryo and a sperm.
Right.
Oh. And now it's, like you said, we could pull back individuals alive during the 80s in today and reintroduce genetic diversity from the past to today. Like, that's sci-fi 50 years ago.
I know. I remember reading that book. I'm sure you guys are reading it as well. But now we have a living evidence of these practices with the Przewalski's horse, a Mongolian horse known as a taki, or the black-footed ferret. And again, these are collaborations too, I might add. So just to backtrack a little bit more, being the alliance that we are the 2 conservation parks. We're 8 centers of conservation found around the world, from Australia to Asia to the Amazon. We're also, collaborating with individuals, with governments, but also it's all our donors too. Everyone together helping out in all these practices. Even a little kid coming to the park or the zoo and spending their day buying a plushie or a little toy, like they're directly helping out in a lot of these conservation practices also. So it's a community effort, for all this work we're doing.
And if y'all want to help at home, we do have a pinned message right now in the chat that you can donate directly to. If you donate, send us a confirmation code. We're doing a big sticker raffle when we get back to the office. And that's another way. Could you talk a little bit about just, what this isn't cheap work to be doing, right? No. There's a lot of costs on this. There's a lot of reliance on collaborations and donors.
Yeah.
How can the individual at home who can't come to the zoo make a difference beyond just a donation?
I love that question. I love that question. No. And to your point, again, I mentioned a little bit of it right now. You can, it's simply supporting your accredited zoo and aquarium. As simple as that. There are accreditations all over the world. We were talking about it before we started the live stream, whether it be AZA here in the United States, outside Latin America, WAZA for the world, but every day is an opportunity. There's something I always say to you guys. So I don't care if you're like the emperor of the world, a kid from the barrio, like everyone has an opportunity every single day to make a positive impact in this world or not. A simple thing of like, maybe you go and you buy something at the grocery store that's palm nut sustainable, right? Because that's a big issue going on. Or watch where you're pouring down the drain. Or for me, it's plastic use, the amount of plastic we're buying and using every single day. There's something positive or not so positive that anyone can do with their lifespan every day, Or if you're too much, again, just come visit us if you can, we do a lot of good work, and we are a nonprofit. So to your point, money does make the world go around at times, we do need that support as well. So again, a big thank you to all the donors, of course, for all the work that we do, right?
Oh yeah. If you think about it, over the 50 years, I've been here like 38, 39 years.
Nice.
And I've always thought it was amazing, but I still learn things, and I'm still realizing things. And one, it just suddenly hit me, maybe around COVID also, that you can't take a break on this collection. The nitrogen the cells have to be in nitrogen all the time. If they're thawed, we can grow them and expand them and refreeze them, but you can't do that with 11,500 individuals. So the commitment of San Diego Zoo Wildlife Alliance to supply the nitrogen all of those 50 years during the economic downturns, during COVID, during everything, when they have to find ways to save money, it's really important. The animal collection is number one, and the plants. And then the frozen zoo, I feel like the commitment to the frozen zoo through the organization is also one of the highest priorities. And it's why it's lasted 50 years. Other collections were started back in the time and they didn't make it
because of various economic reasons.
Yeah.
Can you comment a little for those at home, why liquid nitrogen? Like, why can't we just stick it in a freezer and have it frozen?
Oh, I asked this question too. I thought it was fascinating.
Yeah. So the liquid nitrogen, it keeps them super cold. So the cells are growing and dividing, but we can't keep them. If you kept them in the incubator and they kept dividing, they're going to age just like our skin ages. Eventually, your cells can't divide anymore. The telomeres actually end up shortening, and through every cell division, you're aging. So we need to stop their aging process. And the way to do that is put them in liquid nitrogen with a cryoprotectant. If you just threw them in there, ice crystals would form, and the ice crystals are sharp and they would pierce the membrane and the cells would die. So we put in, it's called dimethyl sulfoxide, DMSO. It used to be back in the day a really high sugar solution. So if you think of like how a popsicle is more of a slushy freeze as compared to an ice cube, they're frozen, but not with these sharp crystals. So anyway, by doing that, we stop the metabolism, and they're just kind of in a state of suspended animation until we pull them back out again.
And the colder temperatures too, on the molecular level, less movement, less damage can be happening. So you're more likely to keep everything in the state in which you found it.
Right. And theoretically, they should be able to last 100 years beyond.
What you saying, like about -320? Was that the number? Or?
Fahrenheit.
Fahrenheit,
yeah.
right? Yeah,
yeah.
That's wild.
And Celsius is -196.
Ooh. Yeah. Just a little cold, a little bit.
Yeah, colder than dry ice.
Do you have a favorite animal in the collection, or a type of animal that is like personally really exciting to you?
I mean, I got one, but I'm curious.
Ahead.
You think about yours. I mean, for me, it's got to be the California condor, you guys. So if your listeners or viewers are able to look at the logo for San Diego Zoo Wildlife Alliance, I got it right here. Probably too small for you to zoom in, but you have a lion, you have a bird, right, and you have a rhino. So the lion represents how the San Diego Zoo Wildlife Alliance got started. It all began with a roar, a lion called Rex the lion. So that's what the statue represents at the entrance of the San Diego Zoo, right? The rhino represents the future conservation work that we can hopefully do to save our disappearing rhino species found all over the world, whether it be the greater one-horned rhino or the white rhino. But my favorite, it's the California condor, you guys. This is, for me, the example of what zoos and aquariums can do, how working together you can actually make a positive impact and bring back a species on the brink of extinction. I mean, we're talking 22 individuals in the 80s, 22 California condors left at one point in our history, and they were going to completely disappear. Zoo Botanical Gardens and a few other partners got together, collected the last remaining individuals of California condors, and now we have close to 500 of those animals now, and half of those are flying free in areas here in the United States and in Mexico. And to me, that's like just the best example of something that feels so daunting and so heartbreaking that we can turn it around simply by it's not too simply, but by working together, right, is the point. So that's for me, the California condor in there. Best story, I think. What about you? Do you have a favorite?
Oh, it has to be the rhino.
Oh, the rhino?
Have you seen my office?
Yes, I've seen The Office.
A lot of rhino statues in my office, little figurines, but every one of those.
My favorite animal.
Oh, really? The
Okay.
rhino?
Yeah, the rhino.
Oh, nice. Not bad.
Yeah. Well, I was hired on a 1-year grant all those years ago to try to get rhino cells to grow better. They were like the amphibian of the day, the cells that we just couldn't get to grow. And so they thought, well, let's hire somebody. And I already had a background in human chromosomes, and they wanted to know the chromosome number of the white rhino was actually wrong at the time. Sumatran rhino was unknown. Javan rhino, we still don't know. But it was really important to freeze these cells so that we could study them and then maybe use them for future uses, like Dr. Benershka said. And so I was able to do that. And today, rhino cells are one of our best growing cells. And I got to do the chromosomes, and they have 82 in the white rhino and greater one-horned rhino, and then 84 in the black rhino. So it's the highest chromosome number at that time that we were working with. That was a lot of chromosomes. And you have to, when you want to look at them, you get the chromosomes out of the cell, and we go through a process that's kind of like magic. But you literally drop the cells on a slide, and the chromosomes have to open out of the cell and spread out enough that you can count them, that they're not overlap, but they're not blown out either.
And so what you're seeing is the classic in the textbook, the X's, right? And
Yes.
then you can count the number of them, and eventually you can ID which one's which, 1, 2, 3, 4.
By building a chromosome map, the karyotype. But in the beginning, you're looking at just this metaphase spread. Everybody who's studying anaphase, metaphase, telophase, all the phases of cell division, we're looking at them in metaphase. But how do you get 84 chromosomes to spread out exactly right? And in the beginning,
on top of each other,
in the beginning
and
they were in the top, and I remember joking that I was going to climb to the roof of the building and drop them down. But we have methods.
Does that help if you're higher up?
We didn't really do that, by the way.
Just within inches. You're
Gotcha.
You know, if they're spreading too much, you can touch the pipette to the slide, and so I was doing kind of some of this.
So like it's not ever written, right? Like you were the one who had to discover even the nuances of this technique.
Well,
right, like for those cell types.
Somewhat, yes, for those cell types. So we build off of what was happening in the human world, as we call it. So human cytogenetics, cell genetics, the study, they're looking at chromosomes. That's where I started, and we start with their methods, and then we tweak it because it doesn't work for all these different species. It's sort of the difference between doctors and veterinarians. There's just a way bigger book of knowledge that you have to know.
Okay, sorry to interrupt you, but I want, because it made me think, especially for all the young listeners right now who are thinking about getting into science, like you took kind of a risk going from the human side to the zoo side, wouldn't you say?
Oh, yeah.
I mean, could you talk a little bit about what that was like for you, going from that to here,
Sure. I was in the human world, and then actually waiting when I moved from Central California down to here, there weren't openings in the human cytogenetics field. So I was actually in immunology in biotech, and that's a pretty secure job. San Diego is one of the biotech hotspots. I think it was the first biotech in San Diego.
So in a way, you were like set, like I'm in San Diego, got my job, I'm good, right? But then you hear about this dude in a zoo somewhere in Safari Park doing something, right?
Yeah, and they said, I was young. It was a long time ago, and they said, "Would you quit your biotech job and come here for 1 year? That's all we can promise you, to do this project." And I was like, yes, absolutely. And at the end of the year, I figured I had the skill set to grow cells, look at chromosomes that can be used in a number of ways. And I became actually the first cell culture technician in the department. And now there's 4 technicians and me. Well, one of them's also our lab manager. So we've grown. And then that's separate from the gamete side, who has also a group of technicians. And most of us, well, all the ones who do the banking and freezing of the cells in the gametes, we're all women.
Could you actually talk a little bit about that, about being a woman in STEM back when you started here versus today? And I mean, you're a role model for many women in STEM. My wife looks up to you, and her dream job is actually her work here one day.
Let's say hi to her. Hi, if you're watching.
But can you just talk a little bit about like a message for the women in the scientific community, or thinking about getting into sciences? Like, what message would you give to them for your experience? And also, your passion still shines through. Like, it's clear you're still curious and loving of this. So how did you keep, the patriarchy, if you will, from stamping down and being able to be the curator of this critical institute?
Oh, wow. Yeah. Well, the first thing I would say to everybody is you can do it, believe in yourself. Like, I think, I had that too growing up in school, and STEM wasn't a big thing then. And they didn't really say, women can do this too, but the jobs were there. And if you could, I had the skill set, you just try it. My dad was a plant pathologist. He had his PhD in plant pathology, studying disease in post-harvest citrus for the Department of Agriculture. So I was around a science field, and I think that gave me confidence that I knew a little bit. But if you don't have that, just try it. I actually came here. The reason I learned about the grant job, the 1-year grant job, was because I came to try to volunteer. I thought, well, I knew they did chromosomes here, and I wanted to learn more about it. So you always want to learn, follow your passion. And I always loved chromosomes ever since high school. My high school biology teacher had us cut out from paper some chromosomes. So anyway, I thought if I came here, I could help them and they could teach me more about other chromosomes. And they're like, hey, you have chromosome skills. How would you like to take this risk and take a job? And I said yes. And I think most of the people on my team have taken risks and left very solid jobs to give it a try.
Yeah, because there's nothing set like we talked about with those. Hey, here's rhino cells. But right, let's go. Like that's
someone's got to be the first, right? You know, got to take the risk. But it's just to reiterate that point, kid. Sometimes you're presented opportunities in life where it's a little bit scary. I have a similar story as well to become the spokesperson here, but the concept of you see an opportunity to take it, to grab it, you know, and take hold of it and go full force with it, especially if you have a passion for it.
That's you have to be passionate, right, and excited about this. And it's like the childhood love of animals, right? We all want to be a marine biologist or
veterinarian
or working in it with like.
I was less about water. I was more the bird rap, but I get what you're saying.
Well, I wanted to work with rhinos too. Like that was like how cool that would have been. And it's like to see that passion still in your eyes as you're talking to us today, like. That I think is really inspirational because there's no jadedness. It's just like there's clear love of what y'all do.
Yeah, and try it, and if it's not right for you, it's not for me. When I got in that's kind of why I was looking to volunteer too. Or my niece started in thought she wanted to be an engineer, and then she got into the classes and she's like, nope. Now she's veering more towards the sciences.
We had a couple of questions from the chat. One I think is really interesting is you mentioned 11,000 plus individuals here. How does one select a new species to add? And how do you select how many individuals to add? What is an ideal number for that? And then what is the threshold for adding something?
Right. So every sample we get is opportunistic. We can never go out and say, hey, that bird isn't represented in the frozen zoo. Can we get a sample? Especially the mammals where they could sample live, like an ear notch. Sometimes when they notch the animals for identification purposes, they would otherwise throw that away. We can take that. It's an opportunity for us to grow cells. So everything's opportunistic, and we wait. And if a sample comes, we can't grow everything that comes our way. It's a small group of cells, like I said, are in the incubator for about a month. So
space is an issue.
Space is an issue. Time is an issue. The cells have to be tended. We don't take off. We'll take a couple days for Christmas and things, but we never are gone. Somebody is here at least every 3 days. So we kind of look at things, and if it isn't in the collection, that becomes really important to add because we want diversity. We want to keep growing that diversity and add new species all of the time. But we don't want just one of everything like the pauli, and we need to collect now before they become more endangered. So we kind of take that into account as well. And how many? Okay, this is funny. Arlene Kumamoto, the first curator of the Frozen Zoo, I'm the second. She said we should collect 6 males and 6 females, and that was her kind of her standard at a minimum, right? Some things we collect more. Every rhino, every Przewalski's horse, every California condor, every gorilla. But she said 6 of each sex for most species would be a good starting point that we could move on and collect other species. So that's what we do still today to a degree, but we'll collect more. But if it's under 6 of each, we keep going and we'll take that.
So you have like a running list, and is it with the collaborations, like people know in the collaborative network, like, if you have the opportunity for this, we really need these particular things?
We do. Other zoos support us, and they'll say, so if they have animals that die, or there's an animal if the veterinarian is doing an exam and they might want to take a sample, most of the times they're asking us to bank them, or it could even be, we work with NOAA and Fish and Wildlife, and they'll say, we think it's really important to bank these species. If we come across one, will you bank it? And we'll do that for sure. Yeah. We have to have special permits.
What is the permitting like for that? So you can't just get stuff sent in.
No, all kinds of permits. We follow the ICUC, I won't remember what that stands for, IACUC, but International Care and Use Committee, I think, for animals. So you protect it. The idea is we never want to harm an animal, and so we have to definitely follow those. And then different species have different permits. There's a marine mammal permit that we have. There's a permit just for manatees alone.
I mean, I would imagine there's one for plants as well. I mean, in that, even
Right.
with new science developments, we're integrating plant species in the frozen zoo now too, which is wicked, you know?
Yeah,
so
can you talk
yeah,
about
it's growing. You know, for instance,
what's
the
that
pollen
project?
for a cycad is an example. Now, I'm not a scientist, but my point is like that knowledge is growing, we're evolving in what we do, and also the acorn for
Right,
oaks,
right. The oaks. So the first, okay, so most plants are preserved through their seeds, and you can dehydrate the seeds. And we have a very well-known seed bank. In fact,
Yeah, separate than frozen zoo, I may
right.
add. So frozen zoo, but then we have our seed bank completely separate from this facility.
Right, we have 6 collections of different. They're very specific. Each one, you know, for example, the frozen zoo is frozen and living. The seed bank is living but not frozen. They're more dehydrated. We have a clinical pathology collection. We have an RNA and DNA collection. These 6 collections all together are known as the Wildlife Biodiversity Bank. But the frozen zoo is the oldest and usually the most well known. So back to seeds, usually you would dehydrate them, but some species you can't dehydrate the seed, like the acorn that you mentioned.
Yeah.
It's too fleshy and you can't really dehydrate it. So what they're finding is it's better to preserve them in liquid nitrogen, not the acorn itself. This is not my area of specialty, but it's something like the root tips or the
like
embryonic tissues as they're starting to grow. They can bank those in liquid nitrogen. So we added the very first plant, the Nuttall scrub oak, to the frozen zoo. And then the gamete team has begun. So I said we do all vertebrates, but the gamete team has been adding invertebrates like the sunflower sea star, which is dying from wasting disease. They've been able to get little embryos. And then the really cool one is the Lord Howe Island stick insect. They got semen, and they had not frozen sperm from an insect before, so they used their methods and used their best judgment, and they were successful on the very first try.
Wow,
it was wicked, especially a species that we thought was extinct in the wild too, but they found a small population off of an island, and then we can help out with that genetic diversity too. So that's why, like, I'm always reminding people why zoos and aquariums are so great these days, especially the accredited ones, right? All the work that we're doing, and it's just constantly growing, and it brings you hope. And that's what I want our donors, our visitors to know, that when they come visit us at the zoo or the safari park, I want them to leave with their heart filled with hope, that know that, especially youngsters these days, I'm doom scrolling my TikToks and I'm seeing, feels like the world is burning, I know, but just when I come here and I talk to colleagues and friends of mine and I hear the stories that we're doing, it makes you feel good, and you know there's something to counter all that you see out going on in the world, you know.
Oh, I have a story on that.
Oh, tell me.
I was in the 90s. I was in Africa they were dehorning the rhinos to hope that they wouldn't be poached.
Yes, yeah.
And that was an opportunity, because everything's opportunistic for us, for me to go in there. It was a different subspecies of black rhino. And here at the zoo, we only had a few, and they were wondering, are they different enough that we should keep them? Should we let them breed, or should we keep them in different habitats? And so that was the question. We were learning this through chromosomes. Always goes back to chromosome. And so I went over there and was out in the field for a month collecting samples. And they thought we would find maybe 60 rhinos. We found 11, and I brought back 11 samples using a field method, and then all the cells, brought them back to the lab where you grow the cells. All 11 worked. But it was really sad to see that the rhinos were disappearing in Zimbabwe so quickly, even in the 90s. And then I came back here, and our rhinos of the same subspecies had an offspring. And I was like, I really, really, really love what we're doing.
No, I mean, we've been in it for me close to 30 years now myself, you know, but it really speaks to how much we love the work that we do. And to your point, it's been over 100 rhino calves have been born here at the San Diego Zoo Safari Park. But also left me with another kind of topic we were talking about prior, just say a concept of a species, you know, what makes a species, and even that definition is growing. You know, you were talking about for breeding purposes, right? That could be a definition for it,
right? Like, if you cross a horse with a donkey, you get a mule, and the mule is infertile. So that's something we really have to be careful here, that we need to know everything we can about the individuals. In nature, they're separated maybe by a river or a mountain range, and they wouldn't have the opportunity to breed. So here we have to be sure that we're respecting the different individual populations, and chromosomes can tell us a lot about that. In fact, horses have 64 chromosomes and
Przewalski's at 66.
They have 66,
right? Yeah.
The domestic horse has 64, and the mule donkey has 62. You cross them, you get 63 chromosomes, and they're infertile. Yeah.
So it's something we have to constantly be defining and thinking about, whether it be for breeding purposes or just geographically, regionally. We need to preserve a certain pocket species, whether it be giant panda or giraffe. So these concepts change.
That's hard too, because ecosystems are constantly changing, whether it be through deforestation.
Yeah.
And so, what's the threshold that you consider? The ancestral environment, and now like grizzly bears and polar bears interacting because of global warming and all this climate change. And so, what was the precursor right beforehand of where you draw the line?
Yeah.
It must, again, so many hats being worn for the maintaining these animals. How many people work at the Frozen Zoo to be able to do this? Because this seems like a mega operation that you'd have to be able to do. But the way you were saying, right, is like it sounds like a pretty small team.
Not very many. We have my team, like I said, is 5. The gamete team is 3. There's a few other people. The adding plants, that would the plant team adds plants. The stem cell team is about 3. So really, I would say under 15 total.
Yeah, so this isn't a factory of people who are just pipetting. It's like very knowledgeable, specialized few individuals who you basically are throwing puzzles at.
Yes.
Yeah.
Yes, puzzles. If you love puzzles, then you love chromosomes.
I am not a puzzle guy.
My PhD, I did a lot of chromosome pairing, and then different species. If you had different regions missing, you'd have like the different mismatches of chromosome pairings.
Yeah, isn't it fun? Puzzles.
It's a blast.
You do the edge pieces first. But like you said, it's not a well-known skill, and that's a problem. And so we want to fix that, because I also said you have to get the samples here quickly. Mm-hmm. Okay, so what if an animal dies in South Africa or Kenya or Brazil or Vietnam? We're not going to be able to get the samples here either because of permits or just time or FedEx delays, and the cells will begin to die. So we are on a program now to establish Frozen Zoo type collections, train people in those regions of biodiversity hotspots so that they can use what we've learned over the past 50 years, and then probably learn more going forward. Their species might be slightly different, but they can bank them right there in their countries.
Yes.
And so we just had a cell culture workshop around, I think it was almost a month ago, a little more than a month ago. And we brought people from different countries, Vietnam, Kenya couldn't make it. She couldn't get a visa. So we're going to do that again. But South Africa, Australia, New Zealand, Brazil, the US, some of the people from Fish and Wildlife, they want to bank in their areas. And so we held a week-long course, and it doesn't teach them everything, but they did everything hands-on. They took skin biopsies from an animal that had died. They processed them. They grew cells. We gave them all our protocols, and now they go back to their countries, and we'll still be a source for them to reach out to us.
It's amazing. And that speaks to the collaborative nature of the alliance that kind of you have to have, right, in order to be able to save. I mean, we're trying to save the planet here, right?
No small thing. Literally just the planet, right?
It's not. I love this. And in biotech, it was more like, wait, this is my domain, and I can't tell people because they might take it and publish it before us. For us, it's like there's so much to do. If anybody's willing